Hi, here we are going to discuss the various characteristics used in taxonomy for classification, or this otherwise these are the criteria used for microbial taxonomy. So in broad, we can categorize them as two characteristics or two criteria. One is the classical characteristics, the other one is the molecular characteristics. Then under classical features, we have morphological, physiological and metabolic, ecological characteristics and genetic data. And under molecular features, we have a comparison of proteins, then nucleic acid base composition like G plus C content and all, then nucleic acid hybridization studies, then nucleic acid sequencing, especially the 16S rRNA sequencing studies. So these are the different criteria used for the study, used for the classification of organisms. And actually these data is used either for phenetic or phylogenetic or for numerical taxonomy, right? So this is the primary step, or this is the part of identification. This is the practical side of taxonomy. All right, so here what a taxonomist do is, or what a scientist do is, they'll perform various experiments to find out all these features, and these features or the data obtained after the experiments are summarized into the taxonomist, and the taxonomist will perform the classification process. So this comprises all those classical characteristics. See, there are morphological features. Actually, this morphology are very comparatively easy to study and analyze. And even though it's morphology, the phenotype, that means the observable character, is usually, we know, every observable characters are dependent on the genotype, right? So it is not only the morphological analysis, but indirectly it gives you a genotypic analysis also. And the different characters under study are like cell size, shape, morphology, the ultrastructural details of bacteria, then the staining properties, the presence of flagella and motility. All those are the different morphological features. Then under the physiological and metabolic characteristic, most of the characters are present or absent because of the presence and absence of a microbial enzyme, and also there will be presence and absence of certain transport proteins. And based on that, we have different criteria, or different components are under study. A number of components are there under this particular head. See, like carbon and nitrogen source, cell wall constituents, energy source, fermentation, etc., etc. You can see a number of characteristics which are under study in physiological and metabolic characteristics. Then we have genetic characteristics. That means purely the study of genetics material, especially the chromosomal gene exchange through processes like transformation, conjugation, and transduction are studied under genetic characteristics. Then finally, ecological characteristics, the ecological nature or the habitat of organism, and not only the habitat, simply the habitat, but also the life cycle patterns and the nutritional status, nutritional the way how the organisms are obtaining the nutrients, and all those things matter, right? So all those are considered under ecological characteristics. All right, so in the next slides we will see each of these characters in a brief way. So first, morphological characteristics, right? I think I'm not supposed to discuss in detail of these morphological characteristics. These are, by use of microscopy, especially electron microscopy, we can study most of the morphological features of a cell, and these features will be collected, and the data is used for the classification. The shape, size, and other morphological features are studied with the help of, when in the by the invent of electron microscopy and other advanced type of microscopy, most of the features are easily observable, right? So these data is a very useful data for the classification of microorganisms. Then the physiological and metabolic characteristics are useful because they are directly related to the nature and activity of microbial enzymes and transport proteins. And in that way, you can say that these enzymes and proteins are indirectly connected with the genes. So this study will help us to locate the genotype also, right? Then that means this analysis is an indirect comparison of microbial genome itself, right? And these are the listed characteristics under the physiological and metabolic characteristic. Like most of these things we might have studied in the previous sessions, like carbon and nitrogen source, cell wall constituents, like whether there is peptidoglycan or not, or there is any other difference. Then energy source, based on the energy used, we have classification of organism. Then the fermentation, then the nutritional types, whether they are saprophytes, whether they are pathogens, whether they are symbiotic. In that way we can categorize. Then luminescence, presence of luminescence, motility, osmotic tolerance, oxygen relationships, that means aerobic, facultative, anaerobe. In that way we can categorize. Then pH, photosynthetic, whether photosynthetic or non-photosynthetic. In based on all these characteristics we can classify organisms. So that's about the physiological or metabolic characteristics. Then the third one is ecological characteristics. There are many ecological features under study, because the environment where the organisms are surviving is important. Not all organisms are able to survive in all the habitats, right? So even very closely related organisms may differ considerably with respect to their ecological features. You can have examples like microorganisms living in various parts of the body in human beings are different from each other, right? That means they are different from each other, and they are different from those organisms or the similar organism which are living in other environments. That means if a particular bacteria living in the intestine of a human and the same kind of organism living in the fresh water will be entirely different. So the ecology matters, right? So some examples of taxonomically important ecological properties are the life cycle patterns. That means whether symbiotic, pathogenic, or saprophytic, that matters. Then the nature of then symbiotic relationship with which of the organism it is in an association. Then whether it's pathogenic, that means which is the type of host. Then requirement for temperature, pH, oxygen, and certain characteristics which already we have seen in the physiological features are coming in this discussion also, right? So this is about the ecology. There are organisms which are surviving in the mountain tops, and there are certain organisms which are living in the deep oceans, right? So these organisms are different, and we use these characteristics, we use their ability to survive at extreme environments, and that feature will be used for classification. All right. Then finally, the genetic analysis. The possibility of a genetic variation and genetic exchange is less in prokaryote because there is no sexual reproduction, right? Genetic analysis is widely used in a sexually reproducing organism, but still in prokaryotes we use certain mechanisms like there are certain gene exchange processes in a prokaryotic system, like transformation, conjugation, and transduction, and these are useful for certain genetic analysis, because the conjugation, whether it be conjugation or other mechanisms like transformation and all, these are usually limited in related organisms, right? A successful conjugation is usually limited in related organisms only. So the conjugation occurs between related microorganisms, not between unrelated or distantly related organisms. That's a rare chance for that. So when you analyze two organisms, and when you say that these two organisms are able to conjugate, or they are able to transfer genetic material through a process called conjugation, then you can also say that these two organisms are closely related. So the same is true for conjugation, transformation and transduction also. There may be intake of the genetic material, but there will not be any successful recombination after transformation. So in all those ways, you can say that the genetic exchange of genetic material will result in certain homologous exchange of chromosomes, and this homologous exchange of chromosome occurs only if and only if these two organisms are related or closely associated, right? Then the presence of plasmids. These are the extracellular genetic material, or extra nuclear genetic material. And the presence and absence of plasmids are also being considered as a genetic parameter for classification, right? So that's about these genetic analysis. Okay, so by that we conclude the classical characteristics. So in short, the classical characteristic consists of the four major things. This is the genetic analysis, the ecological characteristic, the physiological characteristics, and metabolic characteristics, and all. So all those characteristics we have, and also the morphological character we discussed in brief, and you can go for further reference for the for all those things. Thank you for listening.